Hello and welcome to the Archaeobotany seminar. In this first sequence of slides, you will get a very general introduction to the basics of archaeobotany, with a focus on the main research questions this discipline is contributing to, a short overview on botanical methods that help to interpret archaeobotanical data, the different forms of preservation, and the taphonomy of botanical macrofossils. Archaeobotany is a relatively young archaeological discipline that may be considered jointly with zooarchaeology as archaeobiology. The European and American terminology sometimes deviate in relation with a different history of research tradition. In the US, paleoethnobotany is an equivalent for the European archaeobotany, but with a quite different research tradition, being strongly linked to cultural anthropology and ethnobotany, while European archaeobotany was traditionally a branch of botany and associated to the geosciences. However, paleoethnobotany and archaeobotany are both interdisciplinary fields addressing the meaning of biological remains in archaeological contexts. The figure shows how the research questions strongly relate to ancient human culture, whereas the methods are mainly adapted from the natural sciences. Archaeological finds of biological nature are often referred to as ecofacts, in contrast to artifacts which are human-made. Archaeobotany mainly investigates plant remains from archaeological sites, and these studies are often referred to as on-site investigation, whereas the study of organical remains from natural sediments like lakes and bogs, with the goal to address human impact on the environment, are named off-site investigation. The pair of terms on-site and off-site investigation is also used in a different way in archaeology, with on-site describing the excavation and sampling in the field, while off-site is used to name the laboratory analysis. Which chronological range is covered by archaeobotanical research? In general, the whole sequence of the development of humankind, that is starting with the Paleolithic until modern times. The further we go back in time, the more incomplete the archaeobotanical evidence becomes. This is due, on one hand, to decreasing preservation with increasing age of the archaeological layers. On the other hand, it is related to the fact that only with large-scale storage we may expect numerous plant remains. Most archaeobotanical investigations are therefore conducted on material younger than 10,000 years, which is equivalent in time with the beginnings of agriculture. With the emergence of sedentary communities, large-scale storage, and the production of resources, more opportunities for deposition of large amounts of plant remains were given. The major impetus for the beginning of archaeobotanical research were the works by Charles Darwin, and in particular his book published in 1868, The Variations of Plants and Animals under Domestication. During this time, archaeological discoveries revealed excellent preservation of plant remains in Egypt, but also in Central Europe at the lakeside settlements. The Swiss paleontologist and paleobotanist Oswald Heer was the first to investigate plant remains from an archaeological excavation. For more than 100 years, the primary interest in plant remains centered on the evolutionary history of crop species. Archaeobotany's establishment as an academic discipline during the 1960s coincided with the refinement of radiocarbon dating and calibration curves. It focused on subsistence and technology and emphasized human adaptation to the environment as a part of the developing processual archaeology. Growing interest in the role of plants in the development of ancient human societies led to more systematic archaeobotanical sampling at archaeological sites. Meanwhile, archaeobotany is a part of integrative archaeology, which thrives for multiple lines of evidence in reconstructing our past. To understand how archaeobotanical data is interpreted, we need to know the theory behind archaeobotany. Although developed much earlier, archaeobotany became a standard discipline in archaeology only with the development of processual archaeology in the 60s of the 20th century, along with the theoretical concept of processual archaeology, which aimed to search for plausible explanation in archaeology through applying methods from the natural sciences. The linking of physical remains to human behavior and natural processes was described by one of the most influential processualists, Louis Binford, as the Middle Range theory. Archaeobotanical interpretation is based on the principle of uniformity, which uses modern ecological, ethnographic, or experimental observations and data to interpret past conditions and behavior. To give some examples, modern biogeography of plants that describes ecological characteristics of plant species, such as the climate or soil types that are associated or restricting these species, is used to conclude on ancient vegetation and climate conditions. Ethnographic observation in villages with traditional agriculture reveals crop processing strategies that create a specific composition of crop processing byproducts. Archaeobotanical assemblages can be analyzed for their similarity to crop processing stages, thus are interpreted as if ancient people were exactly using the methods observed in the ethnographic example. The principle of uniformity is what geologists and archaeologists apply when reconstructing ancient conditions. In any case, archaeobotanical remains are indirect sources for interpreting cultural behavior. That is, they have to be interpreted before reconstructing diet or an ancient landscape. They represent proxy data for reconstructing the past. Here you see the main areas of contribution in archaeobotany. On the left side you have the major research topics, on the right side the types of plant remains that are usually addressed to answer these questions. The beginnings of cultivation and domestication are related to very early sites from the PBN and the Neolithic. Agricultural technology is mostly focusing on fully developed agricultural societies, that means from the Chalcolithic period onwards. The development of vegetation and species diversity, as well as climate and environmental history, is addressed by the investigation of wild plants, and with vegetation, of course, not only macrofossils can be considered, but also pollen and wood charcoal, in contrast to small seeded plants. And then for climate and the environmental history, wild plants and particularly the distribution of plant species as well as crops may be helping in addressing this question. Additional methods such as the stable carbon isotope analysis may also help to address environmental aspects. As concerns the question of diet, of course, all periods are of relevance here in all kinds of plant remains as well, because most of the plants can, of course, also be used for subsistence. The aspect of cult and ritual is very often not one of a major topic in archaeobotany because it requires specific contexts like graves, sanctuaries, and also there plant assemblages are often not preserved. So most of the main research topics involve an economic component, including the acquisition of resources and food production, as well as the way of use, redistribution, and consumption. Food in archaeology is most important not for its biological aspects, such as intake of vitamins and calories to provide health, but for its reflection of cultural identity. In some cases, food offerings in religious contexts provide even information on ritual behavior. Beside this, an environmental component is frequently involved, mostly through the presence of wild taxa, and enables the reconstruction of human impact on ancient landscapes. Comparisons between sites allow reconstruction of regional differences within a diachronic framework. We will have an extra lesson on the evolution of crop species and the beginnings of agriculture. Therefore, I will only shortly mention some methodological and historical aspects on this topic now. Historically, this kind of research is strongly linked to early evolutionary studies. First detailed biogeographic works go back to the Russian botanist and geneticist Nikolai Vavilov, who discussed diversity centers of modern crops and the phylogeny of cereals. One result of these early biogeographic investigations is our knowledge on the distribution of wild progenitor species of modern crops, which defined the area of the Fertile Crescent in the Near East, as indicated in this figure here. The four main aspects to approach the question of the evolution of crop species were the analysis of variability of crop plants, the identification of wild progenitors, their localization, and the identification of changes relevant to adaptation under domestication. While these were investigated in the past mostly through classical taxonomy by morphological comparison of the plant's anatomies, molecular and biochemical methods are more in use today. Only recently, archaeobotanical research would contribute considerably to the understanding of crop evolution through the morphological study and radiocarbon dating of early Neolithic cereal finds from archaeological sites. Ancient agriculture is of central importance for the understanding of ancient societies, and there are a number of questions that can be addressed by archaeobotanical methods, such as how the Neolithic agriculture dispersed from the Fertile Crescent into other regions, or how mobile were early farmers. Were there social differences between households within the same settlement that indicate any kind of hierarchic structures? Which economic goals did crop production have? So was it more oriented towards basic subsistence, or was there a surplus production? What was the intensity and scale of plant production, and which technologies were used? So do we recognize soil management, manuring, irrigation, extension of fields? And what were the consequences of early agriculture? So, for example, addressing overexploitation or salinization. Of particular importance is the archaeological record, which provides basic evidence on features relevant to the reconstruction of agricultural practice, such as field borders, terraces, dams, drainage systems, irrigation channels, and so forth. Additionally, paleopathological and micromorphological analysis may provide evidence of soil modifications through manuring, salinization, or overexploitation. Food is an important aspect of cultural identity, and therefore the plant component in ancient diet is an important issue in archaeological research. This table here shows how the different processes in food provision are related to cultural factors. So, for example, knowing about the processing of food in a site can inform us about labor division or about the technology of preparation, and through that about the economy of a society. Context-related archaeobotanical sampling can contribute to our understanding of the multidimensional character of food systems, but without further knowledge of the archaeological context, archaeobotanical remains only indirectly reflect ancient peoples' diet, and using their proportions to reconstruct the composition of past diet directly would provide a biased perspective of the food preferences of a site's inhabitants. Archaeobotanists sometimes interpret the use of plant species that are commonly in use today for food by comparison with ethnographic examples. But the only direct archaeobotanical way to know what ancient people have eaten is through the study of stomach contents and coprolites. However, ancient diet is indirectly reflected in food preparation and storage. Wild plants from pre-agrarian contexts are generally interpreted as collected resources, and in the case of fruits, nuts, and tubers, as food plants. Wild progenitor species of modern crops in aceramic Neolithic sites are also generally considered as food plants. Seeds and chaff remains associated with installations such as storage pits or silos, hearths and grinding stones, or plant parts such as phytoliths and starch grains on agricultural tools provide evidence of consumed plant species. Large-scale systematic sampling of occupation layers enables archaeologists to identify activity zones of food production. In agricultural societies, crops indicate the cultivated components of the diet. In rare cases, trade of food plants can be investigated with archaeobotanical methods. While the occurrence of single wheat species of non-regional origin cannot answer the question of whether trade was systematically practiced, large-scale storage of exotic food plants is a positive indicator for long-distance economic relations. Archaeobotany also contributes to the study of social aspects of food consumption, including the role of food in social relations, consumption habits of specific groups according to socially perceived norms and ritual contexts. In general, the archaeobotanical assemblage of ritual contexts differs in composition from contemporary household finds. Roman period burials, for example, often contain accumulation of complete fruits, a characteristic of the funerary ritual. As already said, the most direct way to investigate ancient diet is through the analysis of stomach contents, as has been done on northwestern European bog bodies, mostly from the Iron Age, such as the Grauballeman or the Bronze Age Iceman Ötzi. There are only about 20 known completely preserved bodies in Central Europe. In some cases they seem to have been sacrificed or murdered. Modern methods make use of histology as applied in forensic. The analysis of the highly fragmented tissues, such as cereal bran extracted from the guts, is very time-consuming and often does not lead to species identification. Another problem is the quantification of such highly fragmented objects, and in some cases mathematical models calculating the amount of grain by the surface of cereal bran have been applied. Other archaeological contexts informing about ancient diet are pit latrines, which are evident in Central Europe since Roman times and frequently found since the medieval period. They are evident through signs of roofing, such as post holes at edge points, or through coating of the pit by basketry or wood. There is a specific set of indicators that occurs with excrements in wet preserved sediments. These are small fruit stones, fragments of carpels or apples and pears, large amounts of testa and pericarp fragments, cereal bran and seeds of spices, together with leaves and moss that have been used as toilet paper. Sometimes also intestinal parasites are found and then provide information on human health. Household waste like charcoal, carbonized cereal grains and chaff, nutshells and larger fruit stones that have been thrown into the cesspit are also typical. Archaeobotanists often apply ecological criteria to study wild plant species and in particular weeds, in order to investigate ancient growing conditions for crop species and agricultural technology. But how do we define a weed species? In general, weeds appear continuously or spontaneously in vegetation units that are repeatedly disturbed by humans. They compete with crop species and are therefore undesirable. They are often similar in shape to crop species and thus difficult to separate from the crops by crop processing methods. Where do weeds originate from, and how can we know that a taxon represents an ancient weed? There are two classifications for weeds, the Idocorous and the Anthropocorous weeds. Idocorous weeds are originally local species. Some of them may be epiphytes, that is, they have been unconsciously fostered by humans, for example through tree cutting. Anthropocorous weeds are non-native taxa that have been introduced by humans. Depending on the time of introduction, we differentiate between archaeophytes, which have been brought by prehistoric people, mostly from the area of emerging agriculture in the Near East and the Mediterranean, and the neophytes that have been introduced from overseas areas after 1500 AD. Important for the interpretation of archaeobotanical weed assemblages is the fact that they changed throughout human history. This includes that for earlier times we must expect different species within the crop fields which we would not consider today as weeds. Different agricultural technology would also create a different species spectrum. This would consequently imply totally different competition between those ancient species compared to the modern field flora. Taphonomic factors also play a role, as we will see later. As most plant remains are from accumulations, they are not from the same field, thus don't represent plant communities. We therefore don't know for sure which species were weed species in the past. The classification of ancient weeds mostly comes with comprehensive analysis of, for example, storages that have been only partially processed. Multivariate statistics may reveal regularly occurring wild plant taxa that are associated with specific crop species. In this case, the wild plant is very likely an ancient weed. Which characteristics of weeds are used for archaeobotanical interpretation? There are a number of traditional methods, such as, for example, the ecological indicator values according to Ellenberg. These provide specific values for growing characteristics, such as preferred nitrogen ranges in soils. A methodological problem is that these values are determined on modern competitor characteristics, which cannot be transferred to the past. Another classification tool that has been used is the life form and the duration of life cycles, such as differentiating winter annuals and summer annual species. However, these characteristics may change due to the density of plant populations, so are not necessarily applicable to past assemblages. Another tool that has been used was phytosociology, but also here we know that in modern communities there are distinct weed associations, and as already mentioned, the further back in time we go, the higher the likeliness is that these associations did not exist in the past. A new method that has been used is the so-called FIBS, the functional interpretation of botanical surveys. This describes the fact that plants develop similar anatomical characteristics under specific ecological conditions. As an example, high-growing species with large leaves, few but large-sized stomata are indicative of good water availability. However, also here different ways of interpretation are possible, and additionally we don't know the physiognomy of ancient species. After having considered the main research questions in archaeobotany, we should have a look on some methodological traditions. Archaeobotany at least partially developed from phytogeography, traditionally known as geobotany or also as floristic geobotany. This botanical discipline is occasionally applied to interpret archaeobotanical remains and should therefore be shortly characterized here. Phytogeography is concerned with the geographic distribution of plant species and all its different aspects, that is, the factors that govern individual species ranges, as well as the composition of entire communities and floras. Alexander von Humboldt can be considered as the father of phytogeography. He advocated a quantitative approach to phytogeography that has characterized modern plant geography and was developed into gradients in species diversity. A basic element in floristic phytogeography is the mapping of individual species over a defined region to recognize its range or distribution. In German, floristic phytogeography or floristic geobotany is also called Arealkunde. The examples show qualitative presence data of Arnica montana in Germany or Artemisia campestris in southwestern Germany. The signatures are in most cases indicating the time of observation. Thus the maps contain, beside the information on the floristic provinces, also a historical component and can provide information on changes in species diversity. Depending on the coverage and completeness of the geobotanical recording of individual species, the maps can be on a regional scale or generalized to a supra-regional or global level. The sum of such geobotanical observations and the consideration of the patterns of ranges lead to the determination of floristic zones. The different types and sizes of ranges lead to grouping of species, genera or families into cosmopolites versus endemites. The largest floristic unit describes 6 floristic realms: the Holarctic, the Paleotropic, the Neotropic, the Australis, the Antarctic, and the Capensis. Narrowing down the geographic regions provides a more detailed patterning and represents what is commonly known as a vegetation map. The shape of a range generally informs about the requirements of a species. However, the relation between climate and the border of ranges is not strictly causal, because competition between species also plays a role. These aspects contribute to the subdiscipline of ecological geobotany. Ecological geobotany provides a more precise picture of species distribution and adds to the background information. That is, it can explain why species are growing within definable ranges. In general, there is a genetic manifestation of the ecological constitution of species and genera, which restricts them to certain regions, for example, depending on temperature and precipitation as the main range-determining factors, where they are most abundant and occur with the largest number of species. These are tribal centers, or German Sippenzentren. Further away from the tribal center, the number of species decreases. Species with a small physiological amplitude can grow in different climate regions only through changes of habitats. In German, Biotopwechsel. This fact has been defined as the law of relative habitat constancy, or in German, Gesetz der relativen Standortkonstanz nach Walter, which means that climate change leads to a shift to other habitats in order to keep the original growing conditions. As already mentioned, if we are able to consider older documentation of species ranges, we will have a historical component and are able to reconstruct changes in range distributions. Here, paleobotany introduces the main depth in time in going back to earlier geological eras like the Tertiary. Our knowledge on historical geobotany is, however, very limited. Considering the Holocene, data is mostly provided by palynological investigations of lake sediments. As these are regionally very limited, there are large areas where we do not have any information on vegetation development and where interpolation of models for neighboring areas provides the only information on ancient vegetation. An even more detailed approach to understand the composition of the flora is sinological geobotany, more commonly known as phytosociology. The method goes back to the Swiss botanist Josiah Braun-Blanquet and is mostly developed for and applied in Central Europe. In phytosociology, plant taxa combinations are evaluated that characterize univocally vegetation units. Subtle differences in species composition and structure may point to different abiotic conditions such as soil moisture, light availability, temperature, exposure to prevailing wind directions. Phytosociology is a powerful method to describe population dynamics, that is, patterns of response to disturbance and how the community changes over time. As such, the method has highly predictive powers and has demonstrated to be very useful in nature management issues. In general, the most characteristic species is name-giving to the plant association. To distinguish between similar plant communities dominated by the same species, other important species are included. The basic tool in phytosociology is the relevé, that is the documentation of species and formation of species groups in a given area. These species groups or plant associations are ecologically bound to classifiable environmental aspects. Vegetation units, as understood by phytosociologists, may express largely abstract vegetation concepts. Such conceptual units are called syntaxa and can be set in a hierarchy system called synsystem or syntaxonomical system. A syntaxonomical system is an empirical representation of vegetation of a given territory. This close affinity to a specific territory makes it difficult to transfer the concepts into other geographic regions outside the definition of the syntaxonomical system. Here an example of such a syntaxonomical system. The basic unit is the plant association, which is defined by its characteristic combination of plant taxa, habitat features, physiognomy, the biogeographical area, the role in ecological succession, and other factors. The next higher unit is the alliance, which holds plant associations together based on the character species, and the next higher would be the class. There is considerable criticism on applying phytosociology in archaeobotany, such as that in Central Europe human influence is the main factor for the composition of the flora, as there is no original vegetation anymore. Plant associations would thus represent temporary groups of species, and every landscape change leads to new formation of plant associations. This little excursion into the different fields of geobotany gives us an idea of methodological problems if applied to archaeobotanical data. Using ecological information of identified plant seeds and wood charcoal involves methods of plant ecology, which is generally considered to have changed only very slowly within a range of thousands of years, which is why archaeobotanists apply modern horticology to define ecological characters of archaeobotanical species. Here is a list of questions that may be directly addressed to the archaeobotanical data, contributing to both the understanding of paleo-environment and paleoeconomy. Having some background information on archaeobotanical research questions and major approaches, we should have a look on the types of sediments and archaeological contexts in which plant remains are preserved. In general, we can distinguish between autochthone and allochthonous sediments. Archaeobotanical species are only rarely autochthone, which means that the location of deposition is identical with the ancient habitat of species, sometimes termed as paleobiosynosis. Such conditions are more frequent in natural sediments like lakes. Most archaeobotanical remains represent accumulations, that is, they have been transported by different means from different original locations. They are allochthonous and are sometimes described as thanatosynosis. Most of such sediments are somehow related to human activity. Anthropogenic sediments are often described as cultural layers and contain, beside ecofacts, also artifacts. For interpreting anthropogenic sediments, it is important whether the sedimentation was a short-term event or an accumulation of long duration. Short-term events lead to an assemblage that was not changed after deposition, and is also termed as primary refuse, in German geschlossener Fundkomplex. Long-term accumulations may also have been altered by sediment redeposition and are called secondary refuse, in German Ofenhoff-Hansen-Fundkomplex. This kind of distinction is hypothetical and in many cases impossible to be recognized, except in cases when the archaeological stratification and recognition of the context are unambiguous. Here are some examples of anthropogenic sediments. Archaeobotanical remains generally are found in structure-related deposits, either from deliberate human action, as for example storage of grains in pits, or in disposing of rubbish, or they may result from indirect processes that are not intentional, such as silting of ditches. Negative features like pits, vessels, and so forth can also become filled after their abandonment, what is called a secondary fill. Having some background information on archaeobotanical research questions, major approaches and find situations, we should have a look on the types of plant remains that are preserved in archaeological excavations. Principally all plant organs may be preserved under certain conditions. What is preserved under which conditions is a matter of taphonomy, which is the preservability under specific physical and chemical stress. In the Encyclopedia of Quaternary Sciences, the entry of taphonomy is described as following. Archaeologists use the term taphonomy to explain the processes that lead to the preservation of biological remains. There are two aspects to consider when evaluating archaeological data: structural and depositional evidence. Structures are what people construct. Deposits result either from deliberate human action, as for example storage of grains in pits, or in disposing of rubbish, or they may result from indirect processes that are not intentional by humans, such as silting of ditches. Deposits will contain the vast majority of biological remains that are studied by environmental archaeologists. These remains belong to different categories depending on how they reached the site. The processes by which a site is buried will have a major impact on how well archaeological and biological remains survive. Generally, material will be best preserved when burial is rapid and the energy levels by which the burying medium is deposited are low. A good example is the Roman town of Pompeii, which was very rapidly buried by the eruption of the volcano Vesuvius in 79 AD. But we can find rapid burial also in Neolithic lakeshore and mire settlements in the lowlands surrounding the Alps. The converse of the rapidity-energy correlation is also true. Slow burial and high energies make for poor preservation, and biological preservation is therefore likely to be minimal. The chemical components of the plant and the sedimentation environment determine the preservability of plant remains. Proteins, carbohydrates, and starch, which are components of plant cells, are decomposed within days or weeks. Pectin and cellulose, which are part of the cell membrane, are decomposed within weeks to months, while lignin, which is a wood component and present in hard tissue such as olive or peach stones, is decomposed only within months to years. The different types of plant remains have different preservabilities depending on the sedimentation environment. Here is a scheme of the preservation of organic material under different depositional environments. EH stands for reduction or redox potential and is a measure of the tendency of a biochemical object to acquire electrons and thereby be reduced, in our case destroyed. Reduction potential is measured in millivolt. A high positive EH indicates an environment that favors oxidation reaction. Preservation of plant material from excavations may be different from that in natural sediments. Charring is the most common of all archaeobotanical preservation types. It preserves plant material after it has been subjected to burning or smoldering, transforming the plant parts, particularly the dense or woody parts, from a carbon-based compound to almost pure carbon. Potentially, charred material can be recovered from any archaeological site where fires have burned or to which fire waste has been brought. So most commonly in settlement sites, but also, for example, in incineration graves. The most common type of charred plant remains is wood charcoal, because wood is usually used to fuel fires. In addition, the most frequently charred remains are those resulting from the processing of food plants, like cereal grains, chaff, and the wheat seeds that were accidentally harvested. Morphological change through carbonization can be considerable, and mechanical destruction is common. Thus, identification of carbonized material can be problematic. The species spectra are generally smaller, as with uncarbonized plant remains, because there is increased selection through humans with household activities, and for taxa from natural stands usually are absent in household contexts. Due to these facts, the fine density of plant remains per liter of sediment is usually low, and large sample size is required to produce representative results. Waterlogged plant remains are recovered from archaeological features that contained water when they were dug, such as wells, cesspits, sewers, moats, and ditches. But also whole layers may be preserved under the water table, like in the case of the Circumalpine Neolithic and Bronze Age lake dwellings. Waterlogging has the potential to preserve a much greater range of plant material than where plant material is preserved only by charring or mineralization. Mineralization usually occurs when minerals carried in solution are deposited around plant cell surfaces or within inner voids, effectively encasing the plant nature and plant structure. The most common form of mineralization on archaeological sites is as a result of calcium phosphate precipitated from excrements. Therefore, mineralized remains are commonly found in middens, cesspits, latrines, sewerage systems, and comparable conditions, and help to reconstruct what people were eating. Mineralization by metal oxides such as copper and iron are also possible, although such preservation conditions considerably change the appearance of seeds. Here are some examples of uncarbonized plant remains, as they usually occur in waterlogged sites. Most frequent are the cereal remains, including chaff from crop processing, such as rachis internodes of barley and free threshing wheat, or spikelets of emmer wheat. Wild plants from the wider environment are also common, like seeds of birch or weeds such as this of a catchfly species that grows with linseed crops. Very tiny and highly fragile seed, as this poppy seed, preserve quite well under waterlogged conditions. Seeds of collected fruits occur also frequently, as for example strawberry, blackberry, and raspberry. Here you can see some carbonized remains, as they usually preserve in archaeological sites without waterlogged sediments. As generally in agricultural settlements, we mostly find cereal remains, but also other crop species such as fig, grape, olive, or legumes. A major component in such sites are also the wheat species. Rare forms of preservation are those under completely dry or permafrost conditions, mineralization, and plant impressions. Completely dry conditions are generally possible in deserts or enclosed units, such as the tomb of Tübingen. Important here is the absence of microorganisms, either through the absence of oxygen and or hypoarid conditions. Under permafrost conditions, microbial activity is also virtually absent. Such conditions are provided in the most northern latitudes or in vicinity of glaciers, like in the case of the Tyrolean iceman M.Sc. Another rare preservation form is that through salt, mainly sodium salt and cuprous salt. These are important suppressors to microorganisms. An indirect find are plant impressions of negatives, which are meanwhile irrelevant since standard archaeobotanical investigation is established in most sites. Since we talked about the taphonomy of archaeobotanical assemblages, I would like to additionally mention some other aspects or processes that lead to limited preservation of an original botanical population. The first to mention here is due to a broad range of human activity, which starts with preferred locations for plant collection, as in the example here. Farmers are interested in harvesting their crop fields, whereas the surrounding vegetation hardly finds its entry into the settlement. The mode of plant collection also plays a role. For example, when cereals are harvested directly beneath the ears, the chance for low-growing weeds is very small to become part of the deposited assemblage. The full range of crop processing activity leads to selective deposition of plant material. As a consequence, different crop processing stages can be reconstructed through analyzing the archaeobotanical assemblage. We already talked about biological differences of plant organs that lead to different preservability. The more fragile a plant tissue is, and the lower the lignin component is, the smaller the chance is that the plant part will survive before it becomes carbonized. But also with exposure to fire, chances of preservation differ, because the plant organs should neither be too close nor too far from the fire source. The ideal charring temperature would be between 100 and 200 degrees Celsius with low amounts of oxygen. After charring, the remains must be covered with sediment layers to protect them from mechanical destruction. There is, however, a wide range of sedimentation processes with different influence on the preservability of the plant remains. Only a tiny part of the original population will be deposited in what the archaeologists call a cultural layer. Usually we only subsample cultural layers, thus the selective processes are continued by modern humans investigating these remains. Further destruction of remains can happen through the way we sample and process the samples before they are analyzed under the microscope.